This is a tutorial for Gene2Function. I'll be showing how to search for information about the function of orthologous genes, as well as how to search for gene information associated with different disease terms. So starting out with the gene search, first you're going to choose the species of the gene you're searching, then enter the gene name. Then you're going to choose which filter. In this case, I'm going to be using best matches only. So first you're going to see this table where it's showing you the orthologous genes that were mapped from these different species. There are linkouts to the NCBI gene ID, the species-specific gene ID, and the species-specific database. The next section is showing the diopts scores, and then whether or not each score or each mapping was the best in this direction, and as well as the confidence of each mapping. Next is the number of publications for each gene. If you click on one of these links, again, they're all link outs, and you can see exactly which publications mention each of these. Moving over is the count of different types of Go terms. I'm not going to show all these, but any of these numbers can be clicked on, and they're link outs to the data. And if you have more questions about the specific columns, any of these that have question marks, you can also hold your cursor over to see a more descriptive annotation. Next is the protein interaction, genetic interaction, mine phenotype data. There's mine expression data, mine disruption phenotypes, RNAi cell data, CRISPR cell data, 3D structure, ORF clones, researchers, and then this last column is the protein alignment. So first, for the gene you searched, if you click here, you can see the multiple sequence alignment of the best orthologs. So first is a table showing information about the domains, things like where within the sequence they are, some descriptions, and then an ID of the publication. But if you scroll to the bottom of this page, there is the actual alignment amongst the different species. And then if you're interested in a particular portion, you can enter in the region and use this to highlight and see exactly where that part of the sequence was aligned. Brings you to the top of the page, but if you scroll down, it's now highlighted there. All right, and then next we have, for the orthologs, there's a pairwise alignment of the particular ortholog's protein sequence against the searched gene's protein sequence. And we've got that here. At the bottom, you can see some information about known domains. And then also, this is the raw information from each of the ortholog prediction tools. All right, so now the next section is the Paralog Overview. If you click here, you can see paralogs of the particular gene you searched. These are also predicted from diopt, and once again, it's got all the same columns shown in the previous table with the same kind of link outs. So I won't show those again. The next section is the disease overview. This is showing any disease terms that was associated with the gene that was searched, as well as the PubMed ID. And then finally, the gene annotation overview. We first have the Go Slim terms and the count of each term for each ortholog, and then the full Go terms. If you'd prefer to view this table vertically, there's that option here too. And with all these tables, including the tables that I linked out, you can export them easily with these buttons. All right, so now going back to the home page, I'll show the disease search. So you enter a disease name, and this is going to show you a list of all the disease terms that it was mapped to. If you use this button, you can quickly uncheck or check all the disease terms and then select only the relevant ones. For this example, I'll just select a few. Now you're seeing a list of all the genes that were associated with these disease terms that we chose. Again, there's link outs with the human gene ID, and then if you click here, you can see predicted orthologs. And that covers all the basics. If you still have questions or run into any issues, you can use our bug report form here. Thanks for watching.